Vdj recombination, also known as somatic recombination, is a mechanism of genetic recombination in the early stages of immunoglobulin and T-cell receptors production of the immune system. Vdj recombination takes place in the primary lymphoid tissue. Vdj recombination nearly randomly combines variable, diverse, and joining gene segments in vertebrate lymphocytes, and because of its randomness in choosing different genes, is able to diversely encode proteins to match antigens from bacteria, viruses, parasites, dysfunctional cells such as tumor cells, and pollens. For discoveries concerning Vdj recombination as genetic basis of antibody diversity, Susumu Tonegawa was awarded Nobel Prize in Physiology or Medicine in 1987. Background: human antibody molecules are composed of heavy and light chains with both constant and variable regions that are encoded by genes on three loci. Immunoglobulin heavy locus on chromosome 14, containing gene segments for the immunoglobulin heavy chain, immunoglobulin kappa locus on chromosome 2, containing gene segments for the immunoglobulin light chain, immunoglobulin lambda locus on chromosome 22, containing gene segments for the immunoglobulin light chain. Each heavy chain and light chain gene contains multiple copies of three different types of gene segments for the variable regions of the antibody proteins. For example, the immunoglobulin heavy chain region contains 2 constant gene segments, 44 variable gene segments plus 27 diversity gene segments and 6 joining gene segments. The light chains also possess 2 constant gene segments and numerous V and J gene segments, but do not have D gene segments. DNA rearrangement causes 1 copy of each type of gene segment to go in any given lymphocyte, generating an enormous antibody repertoire. Roughly 3A1011 combinations are possible, although some are removed due to self-reactivity. Most T-cell receptors are composed of an alpha chain and a beta chain. The T-cell receptor genes are similar to immunoglobulin genes in that they too contain multiple V, D and J gene segments in their beta chains that are rearranged during the development of the lymphocyte to provide that cell with a unique antigen receptor. Failure of the cell to create a successful product that does not self-react leads to apoptosis. Autoimmunity is prevented by eliminating lymphocytes that self-react in the thymus by testing them against an array of self-antigens expressed through the function of in immunoglobulins. Heavy chain: in the developing B cell, the first recombination event to occur is between 1D and 1J gene segment of the heavy chain locus. Any DNA between these 2 gene segments is deleted. This DJ recombination is followed by the joining of 1V gene segment, from a region upstream of the newly formed DJ complex, forming a rearranged Vdj gene segment. All other gene segments between V and D segments are now deleted from the cell unregistered trademark S genome. Primary transcript is generated containing the Vdj region of the heavy chain and both the constant mu and delta chains. The primary RNA is processed to add a polyadenylated tail after the Psi-1/4 chain and to remove sequence between the Vdj segment and this constant gene segment. Translation of this mRNA leads to the production of the Ig-1/4 heavy chain protein. Light chain: the kappa and lambda chains of the immunoglobulin light chain loci rearrange in a very similar way, except the light chains lack a D segment. In other words, the first step of recombination for the light chains involves the joining of the V and J chains to give a VJ complex before the addition of the constant chain gene during primary transcription. Translation of the spliced mRNA for either the kappa or lambda chains results in formation of the Ig or Ig light chain protein. Assembly of the Ig-1/4 heavy chain and one of the light chains results in the formation of membrane-bound form of the immunoglobulin IgM that is expressed on the surface of the immature B cell. In T-cell receptors, during thymocyte development, the T-cell receptor chains undergo essentially the same sequence of ordered recombination events as that described for immunoglobulins. D-J recombination occurs first in the I-squared chain of the TCR. This process can involve either the joining of the d^1 gene segment to one of 6 g^1 segments or the joining of the d^2 gene segment to one of 7 g^2 segments. Vdj recombination is followed with V² to d²g² rearrangements. All gene segments between the V² d²g² gene segments in the newly formed complex are deleted and the primary transcript is synthesized that incorporates the constant domain gene. mRNA transcription splices out any intervening sequence and allows translation of the full-length protein for the TCR-Si² chain. The rearrangement of the alpha chain of the TCR follows I² chain rearrangement, and resembles Vdj rearrangement described for egg light chains. The assembly of the I² and I+or- chains results in formation of the I+or- I² TCR that is expressed on a majority of T cells. Mechanism: recombination signal sequences. The regional gene segments are flanked by recombination signal sequences that are recognized by a group of enzymes known collectively as the Vdj recombinase. RSSs are composed of 7 conserved nucleotides that reside next to the gene encoding sequence followed by a spacer followed by a conserved non-namer. The RSSs are present on the 3A unregistered trademark side of the V region and the 5A unregistered trademark side of the J region. These are the sites that will be involved in the joining. Only a pair of dissimilar spacer RSSs are efficiently recombined. This is known as the 12/23 rule of recombination. Vdj recombinase. Vdj recombinase refers to a collection of enzymes, some of which are lymphocyte-specific, and some that are expressed in many cell types. The initial steps of Vdj recombination are carried out by critical lymphocyte-specific enzymes, called recombination activating gene 1 and -2. These enzymes associate with each other to recognize the RSS sequences and induce DNA cleavage at the RSS sequences. This cleavage only takes place on one strand of DNA, which leads to a nucleotide attack and creation of a hairpin loop. Other enzymes of the Vdj recombinase are expressed in multiple cell types and are involved in DNA repair following the activity of RAG1 and RAG2. RAG1 is homologous to transposes. One of these enzymes is called the DNA-dependent protein kinase complex that repairs double-stranded DNA. DNA PK binds to each end of the broken DNA and recruits several other proteins, including Artemis nuclease, exocc4, DNA ligase 4, cernose, and any of several DNA polymerases. DNA PK complexes on each DNA end phosphorylate each other, resulting in activation of Artemis. Artemis then breaks the hairpin loop that was formed by the RAG proteins. Exocc4 and cernose act in concert with DNA PK to align the two DNA ends with each other, and also help to recruit the enzyme, terminal deoxynucleotidal transferase, which adds nucleotides randomly to the ends, providing junctional diversity. DNA polymerases I and I1/4 insert additional nucleotides as needed to make the two ends compatible for joining. Ligase 4 finally links DNA strands on opposite ends of the break to each other, completing the joining process. Because of the variability in the exact position of cleavage of the hairpin loop by Artemis, as well as the random nucleotide addition by terminal deoxynucleotidal transferase, the final DNA sequence, and thus the sequence of the resulting antibody, is highly variable, even when the same two V, D, or J segments are joined. This great diversity allows Vdj recombination to generate antibodies, even to epitopes that neither the organism nor its ancestors have ever previously encountered. References. Further reading: Hartwell L.H., Hood L., Goldberg Mill, Reynolds A.E., Silver L.M., Beers C. Chapter 24, Evolution at the Molecular Level. In Genetics. New York, McGraw-Hill PPA 805/807. ISBN A0-07-299587-4, B, D, J. Recombination. Series, Advances in Experimental Medicine and Biology, Volume 650 Ferrier, Pierre Lands Bioscience 2009, 12, 199 p. ISBN 978-1-4419-0295-5.